Hello everyone, my name is Fee and I'm a research assistant working at the University of Cambridge. In this video, I'll be showing you what I do in labs on a day-to-day basis, so I hope you enjoy. A lot of what I do involves extracting plasmid DNA from bacteria. So here are just some bacteria cultures that I grew overnight and I have centrifuged them already. So first of all, I just discard the supernatant, which is the media that the bacteria have been growing in for a while. Then I use a kit to resuspend the bacteria pellet and also to lyse them using the appropriate buffers. And after I leave them on the bench for a couple of minutes, I then use a neutralizing buffer to perform neutralization. A mini prep involves passing your samples and also a bunch of other buffers through the spin columns, which are these tubes that you can see here. Normally you can do this in the centrifuge by spinning them down a couple of times, but that can get very tedious and laborious, so I just use a vacuum pump for that. After I've washed my silica membrane, I then elute my DNA and collect them in an Eppendorf tube. So normally you just put the elution buffer into the spin column as usual, and then it collects at the bottom of the tube. And I usually like to transfer the eluted DNA into a new Eppendorf. After any sort of DNA extraction, I normally perform a nanodrop to measure the DNA concentration of the sample that I've collected. I first load a blank to equilibrate the machine, and then after it's set itself properly, I then load the samples one by one and then take its measurements. Another typical day in the lab for me would also consist of midi-prepping or maxi-prepping. Midi-prep and maxi-prep are basically the same as mini-prep, which is the experiment that you've seen before, but just at a much larger scale. So instead of growing my bacteria culture at say 5 milliliters in a small tube, I'm growing it here at 400 mils in this large flask. Whilst I'm centrifuging my bacteria culture, which takes around 15 to 20 minutes, I'm just setting up my midi-prep columns in the meantime. And because they're quite small, they might fall through, that's why I'm taping them. After centrifuging, I pour out the supernatant and work on it. Work-on is basically a tablet that I put into anything that contains bacteria so that when I pour it down the sink, everything's already killed and we don't pour anything live. So similarly to the mini prep experiment from before, I will also be resuspending and then lysing my bacteria pellet using the appropriate buffers from the kit that's provided. Lysing, which is this blue buffer that you see here, is actually used to break apart the cell membrane so that the DNA within the cell is able to escape and later be extracted. The process of neutralization actually turns this blue buffer into a white, cloudy, and clumpy mixture, and this is then poured into the columns that you see here. Essentially everything that we don't want, which is the white clumpy stuff, will be stuck and collected in the column, and what we do want, which is the DNA, will be stuck to the silica membrane. After the lysate passes through, we wash the column membrane with a specific wash buffer. Washing the membrane helps sort of clean the column, which helps to also get rid of things like salt and other kinds of impurities. So after that, we transfer the column to a new falcon tube for eluting our DNA. The past few times that we've added buffer to the column, a lot of the waste goes through, but this time we will be eluting our DNA, which means that the DNA, which is the thing that we want, is actually going to be passing through the column. So we need to make sure to collect in a clean tube rather than in a flask where we would essentially get rid of all of our hard work. Something that I also do a lot of in labs is cloning. And normally we clone using a very common type of bacteria called E. coli. So yesterday I've already picked my colonies and then I patched them onto these agar plates. So here I'm just noting which of the colonies have actually grown on different types of plates containing different types of antibiotics. For further verification, I will also be performing a PCR on my bacteria culture. So here I'm just diluting it down with water so that I can later lyse it at about 95 degrees Celsius in a thermocycler machine. Because I have a ton of different samples, I have my bacteria in a 96 well plate. So to just speed up the process a little bit, here I'm just using a multi-channel pipette. Here is my deep 96 well plate, and each well in this plate contains a different bacterial colony. So I'll be transferring my bacteria culture to a separate 96 well plate that I've previously already added with water so that I can dilute the entire mixture down. Because I'll be putting my plate into a thermocycler machine, I need to put a PCR seal on top of it, because if you don't, which I've accidentally done before, all of your samples will evaporate and your work will equate to nothing. I normally just use a pen to press against the seal and also the plate, just so that I know the seal is properly tightened. Whilst I'm waiting for my bacteria culture to lyse, I can set up my PCR reaction. So PCR normally contains primers, your DNA template, Taq polymerase, which helps extend the DNA, and also water. After my PCR, I normally run my samples on a gel, but I was in a rush today, so I didn't have time to film it. But thank you so much for watching, and I'll see you in the next video. Bye!